First of all, what is a protein? A protein is an organic compound made of amino acids joined by peptide bonds, primary building blocks of organisms. Now, why are proteins important, you ask? Well, without proteins, you would die because of the many jobs they do, such as enzymes or biological catalysts. Proteins that act as enzymes speed up chemical reactions such as digestion that would ordinarily take too long without enzymes. Pumps and channels in the plasma membrane. The plasma membranes of cells are already selective, but some things are too large to go through. That's where these proteins come in. They take in and push out things too big to fit through the membrane. Muscles. Proteins are what muscles are primarily made of, and muscles provide motor functions, like walking and making up a very important organ, the heart. DNA replication. The proteins DNA helicase, DNA polymerase, DNA primase, and DNA ligase are responsible for DNA replication. Those are only a few things that proteins do, but there are many more. That brings up the question, how are proteins made? In a series of processes called transcription and translation. First is transcription. Transcription is the process in which mRNA is synthesized from the template DNA. Simply put, where the protein's blueprints come from. It happens in the nucleus of a cell. It starts with a piece of DNA. Then, a protein called RNA polymerase shows up and binds to the DNA at promoters or starter signals. It then unzips the DNA, reads one side, and produces a piece of mRNA, the protein blueprints. The side it reads is the template strand, and the side it doesn't read is the non-template strand. The RNA polymerase reads the nitrogen bases on the DNA. There are 4 nitrogen bases on DNA. They are adenine, thymine, cytosine, and guanine. The RNA polymerase makes the mRNA by matching the DNA's nitrogen base with its complementary nitrogen bases. It goes A to T and C to G, or vice versa. But RNA doesn't have T. Instead, it has uracil. So instead of A to T and T to A, it's A to U and T to A. The RNA polymerase keeps making RNA until it reaches a stop signal. Once it reaches a stop signal, the RNA pops off. Now we have a big old pile of mRNA. But we don't need it all, so it must be edited. Chunks of mRNA are chopped off and thrown out. These pieces are called introns. Now we have the pieces that are left. The pieces left are called exons. They are mushed together to make the final mRNA. This piece of mRNA is shipped out of the nucleus to the next phase of protein synthesis. Translation. Translation is the process in which mRNA attaches to the ribosome and a protein is assembled. It happens in the cytoplasm. The ribosome has 2 parts. They float separately until a piece of mRNA comes along. Then they clamp together on the mRNA. But they don't clamp on all willy-nilly anywhere. No. They clamp on at the start codon. This start codon is also known as AUG. Now, a codon is a 3-base code in DNA or RNA, or 3 nitrogen bases. In the case of RNA, each codon stands for a certain amino acid. Amino acids are carbon compounds joined by peptide bonds. They are the building blocks of proteins. There are 20 different amino acids, and the order of them tells which way the protein bends, and in turn, what job it does. Even just one different amino acid can change the whole shape of the protein. As the ribosome reads the mRNA, tRNA shows up. The tRNA brings with them the amino acid. If the anticodon on the tRNA matches up with the codon the ribosome is reading, it drops off the amino acid and zooms off to get another one. The ribosome keeps reading the mRNA and adding amino acids until it reaches a stop codon, either UAA, UGA, or UAG. Once this happens, the ribosome releases the mRNA and the forming protein. Thus, a new protein is born.